रेडियो खांची 90.4 FM हम सबका रेडियो. Myself, Malay Bharti, Assistant Professor, Department of Botany, Doranda College, Ranchi University. Today in this session, I shall be discussing about a very important topic, that is DNA damage mechanism, which is also part of syllabus of PG semester 2. So at PG semester 2 level, this is very important topic related with DNA repair mechanism also, that I shall be discussing in other session. So today I'll be dealing with DNA damage mechanism. So before starting, I must refer certain books on molecular biology written by P. Arora, that is second revised edition, which is published in 2008, and another book in molecular biology by P.K. Gupta, first edition. And I hope these books will help a lot in preparing this topic. First of all, I'll be dealing with DNA damage, how it is occurring, and how DNA damage differs from mutation. Actually, it should not be confused with mutation. Thereafter, sources of DNA damage and types of DNA damage and their mechanism, and finally ROS formation and their effect. Here ROS is very important thing, that is reactive oxygen species. It has great role and great effect on the body, and it should be discussed within this topic. Now let us begin with DNA damage. Actually what DNA damage is. DNA can be damaged by a variety of chemical and physical agents, but spontaneous changes results from the depurination of purine molecule and deamination of cytosine and bases, other bases, and the formation of pyrimidine dimers. These are the main regions of DNA damage. Now let us see, DNA damage is a persistent problem arising from a variety of disruptive endogenous and exogenous sources, which I shall be discussing in this session. DNA damage is actually any kind of alteration in chemical or physical structure of DNA molecule. So any kind of alteration of their structure, chemical structure, that comes under DNA damage. Such a break in DNA strand or base missing from backbone and different other types of damages, it comes under DNA damage. So any missing bases or DNA strand breaking, that comes under DNA damage. DNA damage occurs in different ways, like DNA molecule gets exposed to ionizing radiation. It may get damaged by UV exposure. So there may be a chemical exposure or chemical exposure which tends to damage the DNA molecule, and also there are internal ways that are persistent within the cell that damage the DNA molecule, like replication errors. So all these ways deals DNA damage in one way or the other, like breaking of the DNA strand, or both, as removes the bases from the DNA strand or dimerizing between the two bases. Actually, DNA damage induces abnormalities in DNA molecule. So DNA is not able to act accordingly or able to do normal function. Here, I must clear that there is no difference between DNA damage and DNA mutation. There is lots of differences between DNA damage and the mutation. So now let us see what are the differences between DNA damage and mutation. DNA damage is distinctly different from mutation, although both are types of error in DNA. DNA damage is an abnormal chemical structure in DNA, while mutation is a change in sequences of standard base pairs. Here we see DNA damage is a kind of physical or chemical damage to DNA molecule, whether as there is something being added or deleted, whereas DNA mutation is attributed to a physical or chemical rather than damage. So here we see it's a change. Or DNA damage, we see it makes DNA molecule non-viable. So when DNA damage occurs, the DNA molecule is rendered useless within the cell, whereas DNA mutation does not make DNA molecule non-viable. It still works, although it may differ from the normal DNA molecule, like it may produce the non-functional protein at the end. In the DNA damage, we see the replication, transcription, and translation is all halted. There is no flow of genetic information. Central dogma is not applicable here, whereas there is DNA damage. Whereas in DNA mutation, there is no halt in these processes. So lots of differences between DNA damage and mutation in their functionality. DNA damage is produced by DNA repair mechanism to fix the issue in DNA molecule which has been damaged by endogenous and exogenous causes. Whereas in DNA mutation, most of the time it evades the DNA repair mechanism and mutation remains in the DNA molecule, but the time it does not evade, but most of the time it evades this repair mechanism. So it remains within the cell. Now let us see what are the sources of DNA damage. Actually, the sources of DNA damage is classified on the basis of their origin into two main types. That is, first is endogenous sources and another is exogenous sources. First, the endogenous sources. Actually, it happens to be a substances or a way which is present in the cell already, which is caused by ROS, that is reactive oxygen species that are derived from metabolite byproducts. Second one is exogenous sources, which are the external elements for the DNA damage such as UV rays, X rays, and gamma rays, etc. So, first, let us see the endogenous sources. Among endogenous sources, the first one is ROS, which stands for reactive oxygen species, the free radicals in biological system. Although these radicals are be made under the influence of exogenous elements like production of ROS by ultraviolet exposure, but still we count them ROS under endogenous sources. Second is replication error. So when the error occurs during replication process, rather base is missing or strand is not ligated, and all these factors lead to DNA damage. Third one is poor repair system. We know we have an inbuilt repair system for DNA damage within our cell. And when this repair system is poor to work properly, it leads to damage with the DNA. So the DNA damaged is not able to get repair again and again because there is a poor repair system present within the cell. Now let us have a look on exogenous sources. We see ultraviolet exposure is the exogenous sources. Other ionizing radiations like X-rays, gamma rays, and other ionizing radiations. Also, the other exogenous sources are hydrolysis or thermal disruption or DNA and intercalating agents also tends to damage DNA at a greater level. Besides these all, there are also external biological agents like viruses which can damage the DNA. So there are some ways by which DNA is damaged. After the sources of DNA damage, let us see what are the types of DNA damage that is done towards the DNA molecule and their repair mechanism. Actually, there are several types like oxidation of DNA bases in the DNA molecule. Then second one is depurination and alkylation of bases. Then another one is hydrolysis of DNA molecule, and one more that is mismatch bases, and uh, fifth one is bulky adduct formation, and another one that is sixth, that is DNA strand breaks by ultraviolet. So these are the different types of the DNA damage, of which oxidation of bases in DNA molecule and hydrolysis of DNA molecule and bulky adduct formation, these are very important mechanism of the DNA damage, which I shall be discussing one by one. So, first of all, let us see how DNA damage occurs by DNA oxidation. That how DNA gets damaged by oxidation. We call this DNA oxidation as oxidative to cause damage to DNA. We know that DNA is double-stranded structure, if you remember, that is made up of nucleotide bases. They are adenine, guanine, cytosine, thymine, plus it has also got a ribose sugar and phosphate group in its structure. The DNA oxidation works on these nucleotide bases, that on adenine, guanine, cytosine and thymine bases. As oxidation takes place on these bases, thereby causing alteration in the structure of DNA. In this process, there is an addition of oxygen to these bases occurs. We see we have a normal DNA molecule. It's the production of ROS, that is reactive oxygen species, that acts as free radicals, and these free radicals, there are free radicals that is called ROS, and it has tendency to oxidize their bases. So once bases gets oxidized, the DNA is said to be damaged. It must be noted here, among all the four bases, the guanine gets easily oxidized than the other three bases, because guanine has got high oxidation potential. Not only guanine, but cytosine also gets oxidized by ROS. So cytosine also gets oxidized more frequently than other two bases. So in the structure of guanine, if you see guanine molecule, it consists of huge pyrimidine and an imidazole ring system. But when these guanine molecules encounters or reacts with ROS, that is reactive oxygen species, also known as free radicals, it gets oxidized by slight change in its structure, which is known as 8-hydroxyguanine. In its imidazole ring, it has added oxygen at two positions, so the molecule is known as oxidized. So in this way, the guanine is oxidized by free radicals, that is ROS, reactive oxygen species. Now let us see how this ROS, reactive oxygen species, acts as a free radical in biological system gets formed within the cell. So first we know ROS, reactive oxygen species, they are called reactive oxygen species, which gives us the concept of that molecule being the oxygen atom content. ROS are formed from stable oxygen atom. In its structure, there are oxygen atom having 4 pair of electrons in a particular arrangement. So this is normal oxygen atom, but when it gets an extra electron, which is unpaired electron, at this time it acts as a free radical, and this free radical is oxygen species and highly reactive, so we call it ROS, reactive oxygen species. Now let us see how it is formed within the cell. In mitochondria, generally during the process of electron transport chain of oxidative phosphorylation, the oxygen that gets reduced to water with addition of 4 electrons, but during this process of reduction of oxygen into water, there is a partial reduction of oxygen within mitochondria during oxidative phosphorylation. It's not always getting partially reducing, but sometime it gets partially reduced. Then at that time, there is production of ROS in the form of peroxide, hydrogen peroxide, and these reacting oxygen species and their reaction products such as hydroxy radicals that are very harmful to cells, and they oxidizes protein and also cause little damage to DNA. So this was all about the first type of DNA damage by oxidation. Now the second damage of DNA, that is hydrolysis of DNA or deamination. This is also called deamination. So hydrolytic DNA damage involves deamination or the total removal of individual bases. Loss of DNA bases known as AP, that is apurinic or apyrimidinic sites can be particularly meeting, and if left unpaired, unrepaired, they can inhibit transcription. So hydrolytic damage may result from the biochemical reactions of various metabolites as well as the overabundance of reactive oxygen species, that is ROS. So in deamination, if a DNA strand with missing purines or deaminated base is not repaired, an erroneous bases sequence may be propagated when the strand serves as a template in the next round of DNA replication. For example, if you see where a cytosine has been converted to uracil by deamination, so the uracil will direct the insertion of an adenine on the opposite strand rather than guanine, the correct bases. So the ultimate result of this now heritable changes in the base sequence may be a significant change in the amino acid sequence, and finally the function of protein. So this was about that how DNA damage occur by deamination or hydrolytic DNA. Now let us see another way, that is DNA damage by bulky adduct formation. So DNA damage may also result from exposure to polycyclic aromatic hydrocarbons, that is PAHs, PAH, that is poly uh, cyclic aromatic hydrocarbon. They are potent, ubiquitous atmospheric pollutants commonly associated with oil, coal, cigarette smoke, and automobile exhaust fumes. A common marker of DNA damage due to PAH, that is polycyclic aromatic hydrocarbon, is benzopyrene diol epoxide. Benzopyrene diol epoxide. We call it BPDE. Benzopyrene diol epoxide. It is found to be very reactive and known to bind covalently to proteins, lipids, and guanine residues of DNA to produce BPDE, that is benzopyrene diol epoxide adducts. So finally, these are the way that how these adducts, BPDE adducts, are formed. If it is left unpaired, BPDE, that is benzopyrene diol epoxide DNA adducts, may lead to permanent mutation, resulting in cell transformation and ultimately tumor development. So by this way, this DNA damage occurs by formation of bulky adduct, that is BPDE DNA adduct, benzopyrene diol epoxide DNA adduct. Now, the next mechanism of DNA damage is by depurination. Depurination that involves the loss of purine bases, either adenine or guanine, by spontaneous hydrolysis of the glycosidic bond that links it to deoxyribose. So this glycosidic bond is intrinsically unstable and is in fact so susceptible to random thermal cleavage that the DNA in a human cell may lose thousands of purine bases every day. So by this way, the loss of purine bases, it is a continuous process per day, and depurination process is involved in damaging the DNA. Now, finally, another method of DNA damage, that is pyrimidine dimer formation. So DNA damage by pyrimidine dimer formation, which occurs by ultraviolet exposure. So ultraviolet and other types of radiation can damage DNA in the form of DNA strand breaks. So this involves a cut in one or both DNA strand. So double strand breaks are especially very dangerous and can be mutagenic, since they can potentially affect the expression of multiple genes. Induced damage can also result in the production of pyrimidine dimer formation by covalent cross-link formation. It occurs in cytosine and thymine residue. So a covalent cross-link occurs between cytosine and thymine residue. The most common pyrimidine dimers are cyclobutane pyrimidine dimers. Cyclobutane pyrimidine dimers, that is also known as CPD, cyclobutane pyrimidine dimers, and pyrimidine 6,4 pyrimidine phosphoproducts 6,4-PP. So CPD and 6,4-PP are the most frequent DNA mutations that is found in phosphorus 53p53 protein in skin, which actually the main causing of the skin cancer. It can, pyrimidine dimers can disrupt polymerase and prevent replication of DNA, and finally it leads to DNA damage. So this was all about that how DNA damage occurs and what were the different mechanism of DNA damage. Now here ROS, you see, it is a very important thing, reactive oxygen species, which plays a very important role in DNA damage and all. So how the formation of ROS occurs, we should know in a short that I am going to discuss about some formation of reactive oxygen species that uh, in our life actually oxygen is very important, but some of these oxygen are very harmful, especially species of oxygen, that is ROS, reactive oxygen species, that are chemically reactive molecule containing oxygen. For example, peroxides, superoxides, hydroxyl radicals, etc. So oxygen molecule, by gaining an electron, it converts into superoxide molecules. So this mutation of superoxide produces hydrogen peroxide. This mutation of superoxide produces hydrogen peroxide. So 2H plus 2O2, it forms H2O2. We know H2O2 and oxygen is evolved. So hydrogen peroxide in turn may be partially reduced to hydroxyl radical, that is OH minus, or fully reduced to water. So basically, there are two types or two methods, we can say, that occur, that is involved in ROS formation, reactive oxygen species formation, that is exogenous and endogenous. So in endogenous pathway of ROS formation, ROS is generated normally in our body during oxidative phosphorylation. The complex 4, in complex 4, the terminal electron acceptor is oxygen, which accepts 4 electron and forms 2 molecule of water. Now, instead of accepting 4 electron, if oxygen molecule accepts 1 electron, then it would convert it into a superoxide radical. So during a normal process in our body, some amount of ROS could be generated. Other steps, like during respiration burst by macrophages, ROS is also generated. So let us see, our body is invaded by a pathogen, that is bacteria. So macrophage and dendritic cell would take up those bacteria. So inside the macrophage, these bacteria have to be killed, and then these macrophages kills this bacteria by means of ROS. So in that respect, ROS is also generated. Let us see, we have an inflammatory sensation during inflammation. ROS is also generated. So basically we have seen that there are mainly three types of ROS generation. Now second, that is exogenous mode of ROS generation. In exogenous pathway, by the ionizing radiation coming from sun can damage our body and can produce a lot of ROS, that is reactive oxygen species, and UV radiation particularly have a lot of harmful effects on our body. Apart from UV radiation, tobacco smoke like drugs and xenobiotics, and due to pollution, ROS could also be generated. Not only pollution, the application of pesticides and consuming food and vegetables affected by pesticides, ROS could be generated in our body. So one such example is which is actually a component of pesticide, but it could generate ROS when someone eats with rotenone treated plant or someone consumes rotenone treated plant. So let's see the harmful effect. So this was the way that how ROS is produced exogenously and endogenously. At last, I should mention some harmful effect of ROS in the body. So inside body, ROS is very harmful. How it is harmful, I'll be dealing. First important harmful effect of ROS is lipid peroxidation. That ROS can damage plasma membrane having a lipid bilayer in its structure, which is damaged by hydroxyl radical. Then ROS can cause mutation in DNA. So ROS mediated DNA damage could be ultimately leads to apoptosis also. So ROS could damage the myelin sheath of neurons also, and thus could reduce the velocity of potential transmission, and thereby it could reduce the neural fraction or neural function. So ROS can cause aging also. It could cause multiple type of diseases also. So there are various harmful effects inside the body that is done by ROS. So this was all about the DNA damage mechanism. In our next session, I'll be dealing with DNA repair mechanism. I hope you like this talk. Thank you. Thank you very much.